is because as you're exciting twice, you're going to let something called cyclic electron flow have a potential to occur. Now, we'll talk about that in just a second, but can we at least agree that photosystem 2 comes before 1? Photosystem 2 excites electrons, just like photosystem 1 excites electrons. 2 comes before 1, and your electron passes through this arrow system. Good? Now, what are the names of these proteins or collectively? And you said it a while ago. So you're looking at the electron transport chain proteins right through this zone. What do you think it transports?
Electrons.
That's sensible. Now in the end, your electrons pass off to this enzyme over here. So I'm going to write an E- on this enzyme, and that means that you have electrons over on the enzyme now. Cool? Now, if I start out with an electron over here, let's say that this cap was my electron. The electron got excited, shimmied over, excited a second time, now stored on that enzyme. The electron is no longer here, it's over there, correct? So I need to replenish the electrons. And I'm going to do that by taking water. And just like oxygen was the final electron acceptor, the formed water product is going to be the first electron donor. Now plants respire O2, so that goes out through the stomata. And you have a buildup of H pluses on the inside. Where does this electron go?
That one goes on your transfer chain.
It goes right there in your first reaction center on photosystem 2 to replenish the electron that was bounced up and moved around. Make sure that you get that this electron that was originally here ends up in the end over here. You have to replenish. What does replenish mean? Like to fill it back up, right? Where do you get the electrons? The splitting of water. Now, splitting of water produces O2, which is a byproduct of plants, and it's what I breathe in. And this also produces H+, or protons. So now you have a bunch of protons inside. And I'll one-up you real quick, and I'll tell you that protons also come from this outer area. So there's H pluses on the outside of the thylakoid, and because of this electron transport chain's movement of electrons, do you see the electron? What's the charge of an electron, somebody? And you see the movement of the electron? It seems to be going downward, right? Where does H plus want to go? To where the electrons are, correct? Do you see how the electrons go down to here? What do you think happens to the H plus because of it? It moves inside. So now you have a lot of H plus inside. What type of gradient have I created with this?
Concentration of an
Electrochemical gradient. What does the chemical aspect of an electrochemical gradient mean?
the concentration.
Okay. There's a bunch of H on the inside of the thylakoid and not much on the outside. What is the chemical part? The distribution of the positive charge, right? There's a lot of positive on the inside, but not a lot of positive on the outside, and positive wants to go where it is not. So for these two reasons, H plus wants to go out. Now there's only one doorway. What's the name of that doorway? We saw it last unit or last chapter. We're seeing it again this chapter. What's the name of my doorway right here? If you don't know that, please, please, please make a concerted effort to know that ATP synthase is the doorway for H pluses. It's a turbine, as your book describes. A turbine that spins is H pluses pass through. So do you see what I've done here? I've taken H pluses and pushed them through. Now as that happens, you're able to take ADP and some inorganic phosphates, combine them so that you go from having a diphosphate to a high energy molecule. Yay? Yay.
What's that little box next to
Good
that?
call. This H plus. More H pluses come through. What do you produce more of? ATP. What is H plus attracted to, somebody? Negatives, electrons. Where is the electron at?
In the
So the H pluses all navigate to this area. And you're taking a molecule of NADP and forming NADPH. So your two products of these light reactions, and if you were looking for a title back at the beginning, I would have said this is the light reactions. But your NADPH and your ATP are your two products.
How many of these?
Good call. I'll let everyone finish up and then we'll talk about how many of each.
Your picture's still on my computer.
That's nothing. I'll do it.
Remember that if you want to redraw the picture, just go to the website.
Yeah.
You can hear my lecture again.
I'm pretty sure
You
there's
have a
pictures in the
website?
book too, so.
Yes, I have a website.
Oh my gosh.
Are you a famous YouTuber?
I know.
No, because no one watches my videos.
I tried to watch your movie and didn't play, and I had to call for Wait, what?
Yeah, that's good.
After the show.
So, let's talk about this real quick. The question was, how many of each do I make? Well, if you think about it, I made one NADPH for every one ATP. I made a 1-to-1 ratio. Now, here's what reality is. Reality is that you need, here's the need down here, about 3 ATP to every 2 NADPH. So that's what your real need is. Now you're like, well, who needs this? A plant. Because this is a plant like situation we got going here. A 3-to-2 ratio, 3 ATP to 2 NADPH. Yay?
Yeah.
Good with understanding this. Now the problem that exists is this idea or this problem of I got 3 ATP that I need, I got 3 NADPHs that I need, but my reaction is that strong produces a 1-to-1 ratio, not a 3-to-2 ratio. So watch this real quick. What I have drawn out in black here, and I'll redraw it in black real quick, is something called linear electron flow. Watch my pen move my electrons. You ready? The electron was initially right here, correct? Linear electron flow. What does linear mean? In a line, right? Not reversing like vector in the same direction. Looks like it kind of just went from beginning to end there. Linear is what I'm saying. Oh, I'll get it. Good? Now here's reality. Something called cyclic electron flow. Basketball Season's about to start up, right? Does anyone here watch basketball? 2 people. Great. Has anyone in here not watched basketball at least? Ever? Ever? Okay, at the beginning of the game, they threw the ball in the air. The jump ball, right? Is that what it's called? Whatever. The tip-off. Whatever, guys. It doesn't matter. The tip-off. Now, I want you to imagine the referee there. The referee's supposed to throw the ball straight up in the air, correct? Yeah, right. That probably doesn't happen. If you think about it, anytime you throw something up in the air, assuming that you can't go perfectly vertical, there's a 50% chance it goes a little bit to the left and a 50% chance it goes a little bit to the right. Make sense? I'm going to point to 2 proteins real quick. This protein here and this protein here. When this electron is excited in the photosystem 1, there's a 50% chance that it's excited and thrown a little bit to your left, and there's a 50% chance that it's excited and thrown a little bit to your right. That means half the time the electron goes a little bit backwards and half the time it goes frontwards. Now, your book describes this as something called cyclic electron flow. And what this states is that the electron passes from photosystem 2, excitation, to photosystem 1, excitation. There's a chance, a 50% chance, that this electron is excited in photosystem 1 and passed on to an electron transport chain that it's already been through. Then you're like, you're making this up. Nah, dude. It's in your book. It's called cyclic electron flow. Good question. Now, at least agree with me. Is I do this cyclic form. Let's say I went cyclic and I couldn't get out of it. So round and round we go. Am I ever making NADPH doing this? No. Because NADPH is made way over here. What am I bringing in, though, more so doing cyclic flow? More H+. More H+, more electrochemical gradient. More gradient, more ATP. And don't you need a little bit more ATP for every NADPH? In fact, you need a 3-to-2 ratio, right? Let's take 2 electrons and let's put them through. Here's going to be my first electron. And since I drink too many Diet Dr. Peppers, here's my second electron. I have 2. Yay. Here's my first one. I want you guys to count with me how many ATP I've made. How many ATP did I make? 1. How many NADPHs did I make? 1. Let's kind of do a reversal. Let's DVR this one. TiVo. Back to right here. When it was excited the second time, do you see how it went this way? 50% chance it's going to go this route. 1 ATP, 1 NADPH, right? Here's my second electron. Cyclic flow. 50% chance to the left and right. How many NADPHs have I made now? 2. 2 NADPHs. How many ATPs have I made? 3. Good. Now, that's the way that works out. If you were going to engineer it, you'd engineer it the same way. It's 50% chance, and think about it. If I was to flip a coin infinite number of times, it should work out that it's 50% heads, 50% tails. So with the bouncing up of billions and billions of electrons in a second, it's going to work out that in the end that since you do cyclic half the time and linear half the time, you're going to have about 3-to-2 ratio. It works out perfectly. Cool? Not bad, right? A little bit? Not so much? We're okay? Okay. Sweet. So let's talk a little bit about like what we've done here, because I think what we've done is something like pretty important. So what I'm going to do is I'm going to print out another picture for you real quick, and I'm going to get you guys to draw this one out. Let's take a couple seconds and draw this out real quick, and then we'll talk about it. I guess. Okay, so we have right here, we have a picture of a little chloroplast, right? And hopefully we're getting out, or we're seeing that there are two like integral pieces of this chloroplast situation. We've got this piece initially, like we have the first part of the reaction, which is going to be the light reactions. And then we have over here the Calvin cycle. A lot of people say, well, isn't that the dark reactions? Look, I don't even know if they were ever called the dark reactions, but I know if they were called the dark reactions, I wouldn't stress it. That was never a good name to give them any which way. Because what the dark reactions are saying is they're saying that you could do this in the dark, which is true, but one thing hopefully you're getting is that the light reactions are powering the Calvin cycle. So if you don't have light reaction stuff going on, then you can't power the Calvin cycle for any lengthy period of time. Make sense? So we've got to have a fair amount of light reaction like things given off to your Calvin cycle to even make them possible. Cool? Now what two things are the Calvin cycle, or what two things is the Calvin cycle receiving from your light reactions? So I'm going to write an ATP here. I'm going to write NADPH here. And then I'm going to give it some numbers to be a little bit more specific. How many numbers should I give for the NADPH? About 2?
Yeah.
And how many ATP? That 3-to-2 ratio that's figured out by cyclic electron flow. And what structure am I in here overall? What is my big picture showing you? And how many membranes does it have? What theory are we talking about there? Endosymbiosis or endosymbiotic theory takes care of how there could be 2 membranes. If you don't remember endosymbiotic theory, glossary that one tonight. Endosymbiotic theory, because I'm going to put it on the test, next test, about 7 times.
Awesome. Thanks.
You're welcome. Now these light reactions. How are we building ATP and NADPH? Well, we built our ATP by creating an electrochemical gradient in the thylakoid. We created the NADPH by H's and electrons. H's and electrons. Where would I ever get my H's and my electrons from? Water. I break down water, and that releases O2 in the process. So, I'm going to put an H2O up top. I'm going to put an O2 at the bottom. Y'all can't see that, I know, because this is way too dark. H2O comes in, O2 goes out. And what I'm missing along the way from H2O to O2 is I'm missing a couple of electrons. I'm missing a couple of H pluses. Now, I could only ever make these 2 molecules if I excited my electrons and got the ball rolling with this. So what is it that comes in and excites my electrons? Photons, or light specifically. So my photons come in and they strike my thylakoid, and that's what gets my thylakoid going essentially. Now that takes care of our light reaction needs. Let's come over here to the Calvin cycle, which is the rest of the day essentially. The Calvin cycle is basically a reverse of glycolysis. To reverse glycolysis. Actually, more specifically, I think I misspoke. It would be to reverse the Krebs cycle. Remember what happens in the Krebs cycle, right? You produce CO2s from the breakdown of the acetyl coenzyme A's, or the pyruvate derivative. So you are taking your sugar, or what's left of it, and you're breaking it down to little CO2s. In Krebs cycle, you're going down the steps. So the Calvin cycle would be taking us up the steps. Now, going down the steps, like Krebs cycle, produces NADH and it produces ATP. So going up the steps, it would require energy in those 2 forms. So what do we put into the Calvin cycle?
CO2.
Put in CO2s, and out of it we get
Was it like G3P?
we get G3P or BIP or glucose or sucrose. Depending on the plant, you get some type of a sugar is your outcome.
Can I ask another stupid question?
There's no stupid questions. Please ask.
So you know for cellular respiration, how we break down glucose, it's possible to break down